Hi guys, this is Dr. Gonzalez, and I have a really short presentation here for you about causal claims as they are related to our experimental research. So we're going to talk about making and investigating causal claims. Part of this video relates to internal reliability, which I discussed in a separate video about the 4 major validities, and we touched on internal validity. So this is just a little extension of that so that you can really understand how internal validity and experiments go together. So in chapter 3 of Morling's book, she introduces 3 types of claims. We talk about frequency claims, where we are describing a rate on a single variable, and one of the examples that she provided in the book was something about 20% of people believing that COVID-19 vaccine contains a microchip. And I can easily identify this as a frequency claim because it uses a percentage and it only has one variable or one thing of interest. And here that thing of interest is vaccines containing a microchip. An association claim is what we study in Psychology 4370. These are claims that argue that one level of a variable is associated or correlates with or changes in regard to another variable. So an example would be something like making the claim that regular coffee consumption is associated with better memory. Causal claims are what we're getting at in experimental research, and they argue that one variable is directly responsible for changing another variable. And your book gives the example in chapter 3 of family meals curb teen eating disorders. So the idea is that if a teen eats meals with their family, they're going to have lower rates of eating disorders, and they're actually claiming that the reason is because you eat with your family. Now, causal claims are the boldest kind of claim that you can make as a scientist. They deal specifically with interventions and treatments. Experiments are the only way to prove causal claims. So let's talk about how those experiments do that. Covariance is something that is established in an experiment, and it's a statistical measure that indicates how 2 variables change together. Now, covariance can be established with correlation, but in an experiment, we take it to another level. We're going to manipulate that independent variable and set levels for it. And we want to look at something that might be called the dose response. So in other words, those distinct levels are going to show a covariance in my measured dependent variable. And those different levels are going to be associated with different levels in the dependent variable. There's going to be a related response to it. Now, in an experiment, you use those different levels of the independent variable. We also usually include a comparison group, and that independent variable there can answer the compared to what question. So if your results find that there's no covariance there, we would see that there's no difference between groups, and since there's no difference, there can't be a causal relationship that you can find. So in your experiments, there are many different ways to define the levels of your independent variable. You can have a treatment group and a control group. And in that treatment group, you may not have any levels. So we could explore the relationship causally between caffeine and sleepiness. I have a treatment group that's going to take coffee or an energy drink and then report their sleepiness. And I have a control group that's going to drink water and then report their sleepiness. So I have a level right there. I have that independent variable. It's not broken down into different levels this time, but it is still set up to where I can make a comparison because I have a control group that is not receiving that treatment variable. Another way you can define it is having a treatment group and a placebo group. So say I want to test a new sleep medication. I have a treatment group that receives 5 milligrams of that experimental drug, and then I have a placebo group that's going to receive the placebo, which is also called a sugar pill. Another way that you can define it is by having treatment groups, plural, and comparison groups. So I could ask the question, is note-taking by hand or laptop better for recalling information? So both groups are going to be considered treatment groups because there is no control group that's not taking notes. Now, do you only need 2 groups? The answer is absolutely no. In a drug testing situation, we may want to compare different milligrams of that drug. In the note-taking situation, we could adapt it and have laptop by hand, and then we could just have some participants that sit and listen to the lecture. I can also have one group that has multiple levels. And when we have within group designs, your participant is your own comparison group. So I went over within group designs in the experimental design video, and we're going to talk more in depth about what that means there. But in that group, you may give a pretest to your participants. Maybe you're asking them to recall words after giving them a list of words after they've been sleep deprived. So maybe I give them a baseline test beforehand, a pretest. When they're not sleep deprived, I record their data. Then I bring them back 5 days later. I put them in the sleep lab. I deprive them of sleep for 8 hours. And then I give them the same test. So I've already got my comparison group there with them because it's a within group design. We've talked about in a separate video how experiments and internal validity are related. Well-designed experiments are going to establish internal validity. And in Chapter 3, Moreling went into the specific threats to internal validity that can exist. And those are design confounds, selection effects, and order effects. So we're going to talk about those briefly. So design confounds occur when you have a second variable that unintentionally varies systematically. That means there's a pattern there along with your independent variable. So, for example, you've got one group in a study that receives a treatment in a well-lit room, and the other group happens to be in a room that is dimly lit. Lighting now becomes a confounding variable because whether or not you like it, it could influence your outcome, and it can make it unclear if it was the lights or the independent variable that caused your results. So what it does in the impact to internal validity is it threatens your ability to make a causal conclusion because you've now introduced another alternative explanation. So what we need to do here as a solution is we need to hold all the other variables constant, use random assignment, and standardized procedures. So in that particular example up there, maybe I want to fix this to where I bring my treatment group in that well-lit room, and I do what I've got to do with them. And then I bring the other group in, and I do the same thing, but I'm using the same room. I also want to make sure that any procedures that I'm using are standardized as well. Selection effects happen when you have participants in different groups and they're systematically different from one another from the beginning of your study. So maybe you have some participants that are choosing their own group. So maybe you've got some morning people in one group and night owls in another. That pre-existing morning versus night owl could be a problem in a sleep study, and it might influence your outcome. So my solution would be to screen for chronotype. That's what that is. Morning people and night owls is called sleep chronotype. I want to screen for that, and then I'm going to use random assignment to equally distribute my night owls and my morning people in their groups so that they have an equal chance of being in both the treatment or the control condition. These are just a couple of examples here that are showing you selection effects. Here's you've got this nice distribution of people with 5 green and 5 yellow. And when I use random assignment, I'm going to have 3 green, 3 yellow, 3 green, 3 yellow. Over here, I have something called matched pairs. So I'm going to pick my 2 participants with the highest GPA, then the next 2, and so on. And those are a way that I can help control for selection effects. Order effects happen when your participants experience multiple conditions in a sequence, and the order that they experience those conditions actually influences their outcome. So, for example, if a participant takes a memory test after a caffeine condition, and then again after a no caffeine condition, The practice effect from the first test could actually skew the second result. So, in other words, they simply got better on the test. So, maybe I need to do something to prevent that from happening. I might need to change my study design and maybe not use the same person in both conditions. This might be a situation that would call for two different groups, not repeated measures. We could also use counterbalancing. That's where you vary the order of conditions across participants, and that might control for those effects as well. I know this is a really short video, but please make sure you check Canvas for any assignments, templates, or instructions for this module's assignment. And please remember to reach out to me if you have any questions or concerns. Take care.